Now, looking at the setup, we are using it to demonstrating osmosis using a non-living material. So the non-living material that we are using as a semi-permeable membrane now is cellophane bag. So we fill it with water, and we fill the beaker, or we put a salt solution in the beaker and mark, because the beaker is graduated. So assuming that this is our beaker, which is graduated, and if this is 40 ml of water, of salt solution, and we suspend this one, that is the salt, we suspend the water inside the cellophane bag and put it there and leave it for some time. After a while, it will be discovered that water molecules will have moved from the weak solution that is inside the cellophane into the salt solution in the beaker. Why? Because this is the weak solution, whereas this one is the strong salt solution. So water molecules will move from regions of high solute concentration, but because this is where we have weak solution, the water molecules will move out. We have weak solution here, and we have strong solution out. The movement of water will have. So the increase in the salt solution due to influx of water molecules is what will, prove that osmosis has taken place. Now, we also can demonstrate osmosis, using a living tissue. As we have said earlier on, demonstrating osmosis using a living tissue can demonstrating osmosis using a living tissue. Demonstrating osmosis using a living tissue. Generally, the tissue that we use could be peeled yam tuber, could be peeled pawpaw fruit, and it could be pig's bladder. If it is pig's bladder, we can use it the way we use a cellophane paper. But if it is a peeled tuber of yam, what we do is we just cut a portion of the yam tuber and make a cavity in the peeled yam tuber. Make a cavity, make sure that the cavity is thin enough to allow for movement of water molecules. So you make a cavity in the yam tuber. And after which you place it inside the bowl. At this point, you have this one with open end. You put strong solution here and mark where it is. And here in the bowl or basin, you put a weak solution. So this one is a basin. This one is our peeled yam tuber, which is the living tissue. Here we have a weak, maybe salt solution. And this one is our strong salt solution. And you set it up and leave it for some time, for hours, for like 4 hours. After hours, you will discover that osmosis will take place. Water molecules will move from regions of lower concentration. Because this is weak solution, we expect that water molecules will move through this permeable membrane into the cavity, or into the strong solution in the cavity. And that will cause increase in the volume of solution within the cavity, so that at the end of the experiment, the level of solution must have reached from point A, it could have come to point B. So if it has come to point B, it shows that osmosis has taken place. Now, how do you get your weak solution and the strong salt solution here? The difference between weak solution and strong solution is that the sodium concentration is high in one, and the sodium concentration is low in one. If I have 2 beakers, and in these 2 containers, I have 50 ml of water. This is also 50 ml of water. If I want to make this one to be weak and this one to be strong, all I need to do is to dissolve 5 grams salt in this one and dissolve maybe 30 grams salt in this one. So if it is 5 grams of salt that are dissolved in this 50 ml of water, and I dissolve 30 grams of salt in this 50 ml of water, this is a strong solution and this is a weak solution. So in that wise, what you bring into the cavity of the living tissue is the strong solution, and you put the weak solution inside the basin and now place it and make sure it doesn't cover the tissue up. So through this, you can show and demonstrate that osmosis takes place in living tissue. So through this, we have been able to make points, notable points on osmosis. In your next classes, we'll be able to look at other applications of osmosis as it has to do with plants and animal cells. Before we go, I would like to give you assignments. I'd like to give an assignment. section of the assignment. Assignment. Now, what you do is to tabulate 4 differences between osmosis and diffusion. And you have to submit this assignment within the next 7 days from here. So I expect that on the 15th, submit latest by 15th of May 2020. And how do you submit? Just prepare your answer and upload to my email. I will access it. So you submit to my email. It's written here. enochdo24@gmail.com. So write your name on your answer. All you need to do is just type your name on top of your answer and upload on my email. Good day to you. We'll meet in the next class.